Hi, can you hear me?
Yes, sir.
Okay, thank you very much. Okay, I would like to start again the first lecture, the workshop in the fundamental brain-inspired robotics, focusing on this lecture on the rhythm and memory. I would like to start from beginning again. Okay, it's a kind of summary. We discussed the previous lecture. Oh, this is a squid. We can get giant axon from squid. We can investigate such an electronical circuit activity or property from this tiny but big the neuron in the human brain, giant axon. Yes, real squid. I really respect the professor Gen Matsumoto, his very world famous researcher scientist. He's gone. He discovered some specific chaos activity in the giant squid axon. But he also the famous person. He invented clean system in the world of the squid. All time, people do not know why squid is dead in the artificial condition. Finally, he found the mystery for this. Then ammonia is increasing and in some specific level. Then finally they did in the artificial environment. Then the Professor Gen Matsumoto developed the cleaning system. Then now, I'm not sure that in the world, but then in Japan, so many the restaurant can the keep the living squid in the inside of the restaurant. This is a very funny but and very important technique. Okay, we are discussing the fish brain, very tiny but very sophisticated. Even such a tiny brain, they can think about taking care of their children, putting water again and again, preventing drying condition of the egg until hatched. So, if according to the effort, the evolution, the long time, the mammalian brain developed like this. We can investigate the large the mouse instead of the human brain. So many medicine can be tested in the mammalian brain, like a rat and the mouse. So therefore, we notice the similarity of the structure, the brain function of the rat. So this is the summary, types of memory in the brain. We know short-term memory or working memory, long-term memory. Long-term memory also they classify two types, explicit memory and implicit memory. Or let's say declarative or non-declarative. Explicit memory have two types of the memory. One is episodic, to record event, what you did, what you ate, where did you went. Semantic memory, the fact, okay? There is some Japan, Tokyo is the capital place, 1 + 1 = 2. This is a fact. Implicit memory is related to motor learning function. We can drive the car or the riding in the bicycle. Yes, we can behave, we can control how we can drive or riding in the bicycle by our body, but you cannot explain how you can do, okay? It's implicit memory. Implicit memory is classified into multiple types, at least procedural and perceptual representational. Procedural memory is like a reinforcement learning. Let's say skill, simple conditioning is a part of the procedural memory. This is a famous function of the hippocampus. Hippocampus is the one specific portion of the brain human and rat commonly has. This is the first time you put the mouse in the pot. Mouse doesn't like to walk the same in the water. The mouse is trying to find some escaping point or some prey. But white paper, the water prevent to recognize something inside the water. This mouse try to find some place, but finally and occasionally find some places. Oh, now this mouse is relaxed. Oh, lucky. This is a natural action. You can remember if you're staying, it comes to the novel place. Okay, if you know, you don't know about the location, you have to wandering into the route, how you have to go. Destination unclear. That's why wandering and wandering again, like this behavior. Okay. The rat is put in the pot, they are wandering. But repeatedly, same procedure done in the same rat, the mouse. After 5 trial, this rat is smoothly located at the move to the such a plate very quickly because of the memory. This is called spatial memory. There's some specific cue, video camera set at the top, and the monitoring and the behavior. Some landmark is presented in some places. Then rat can see some such a the node. This is actual function of the hippocampus. That's why hippocampus is necessary to record such as episodic memory. If hippocampus removed from the brain, they cannot find the places. Therefore, control means the normal mouse. If trials repeatedly done, latency is the time to reach the plate decreases quickly. It means, okay, you know that the mouse knows the place and even the different location. Even if the starting point are different, they can reach quickly in some specific position. This is the function of the spatial memory or map. Map representation exists the brain according to experiences. Therefore, yes, normal mouse try to find some specific place, hidden platform, difficult to find at glance. Finally, it reaches. After 5-10 sessions, mouse can reach quickly. This is the power of the spatial memory. But after removal of the hippocampus, specific brain portion, they never reach the place in the same way. Just wandering again, then reach procedural way. This is also strategy. That's why you know dementia Okay, elderly person cannot remember their own memory or what they are going to, the name of the friend, the family, but they can behave because of the different function of the brain. This is sound of the neuron spike. Okay. You saw the giant axon action potential. It's a pulse. The pulse generated the single neuron. If you recorded 100 neuron, such a spike occurred repeatedly like this. Okay, this is coming from the paper, the Skaggs and the McNaughton's group in the University of Arizona. It was they are collaborate in the past in my boss in the Brain Decay Institute. So they recorded such a specific pulse. If you obtain the summation of the pulse, okay, you can see some specific tendency, spike activity, and goes up and down, goes up and down. Like a concert master, conductor of orchestra. Yes, this is a kind of orchestration. Neurons fire synchronously up and down according to rhythm. It represents hippocampus theta rhythm is 8 to 10 Hz exists during the rat is moving in some specific places. Like a music. Music is generated from hippocampus because of the synchronization of the neuron activity like this. So then this is a special electrode putting inside the brain as a needle and then get such an electronic voltage. The group of the Bruce McNaughton, they published the Science of Nature repeatedly. Very productive laboratory. It's what I was so surprised visiting his laboratory in the United States. So by the way, this is some specific machine to record the brain the activity inside the hippocampus. Then also they record behavior because of the LED the highlighting their position. Then rat is such a needle, the putting inside the rat, and then special surgery. They can behave, but also we can record the actual live broadcasting of the neurons firing like this. So this is a very astonishing the technology. They proved brain activity can be recorded. Plasticity, change of the neural network is happening during such a living behavior. Then they provided such a specific machine to record behavior plasticity both together and online. This is a way of the how they can record neural net activity different way. This is a 50 micrometers massive the dense the network the neurons exist. You need to put the needle inside and different the electric activity is recorded mutually. This is very interesting the phenomenon, and then because of the position of the needles, and then even the different activity receive the single electrode, but the property are different. Therefore, such a clustering of the data may work well. Then recognize different cell activity. Okay, this is quite interesting. The method, and you may know the tactile party effect. You can talk your friend in the party, but then you can also hear some other voice, but then you can concentrate some specific person's voice, not because of the cross. Okay, this is the kind of elimination of the brain function too, or independent component analysis or some other mathematical algorithm helps to make clustering, differentiate different property of sound. That's why image processing, the auditory processing is possible to discriminate different sources. We are using similar kind of technology in the brainwave, the recording. EEG, electrography, it's also contaminated. So many signals contaminated in one single channel, but we can isolate it, the different type of signal according to the property. So my work in my PhD season, I focusing in the such a how cognitive map is generated in the hippocampus. So this is the phenomenon. Different neurons represent different portion, which means the rat is locating some specific portion, these cells fire. If rat is green portion, this green cell fire. Different neuron has a different role. They fire differently, depending on the location of that. This is a quite amazing things because traditionally in the neuroscience, neuron react according to input. Okay, this is more natural sensitivity. But in this case, neurons fire depending the place. Place means you can see forward, backward, right, left. It's a different stimulus and signal. Even if the different stimulus is coming, this cells consistent fire depending location. That was the kind of amazing things. That's why this is not simply reflect of the input oriented. Network represent the spatial position. It was a big mystery. I tackled with this issue, how this circuit is generated, specifically from the hippocampus. This is an organization, enteral cortex, receive so many information outside, CSV as recurrent network, like the mutual connection in the new network. And CA1 is our output. So how these networks can be finely organized is worth a big question. This is systematic. Okay, my supervisor, the PhD, Dr. Yoko Yamaguchi, is focusing on such a mathematical aspect, focusing on theta phase precision. It's very systematic firing activity. I showed orchestral rate, very nice sound you hear. Similarly, hippocampus shows that the rhythm, like a conductor. Conductor, the plays, okay, 1, 2, 1, 2, 1, 2, 1, 2. This is a kind of global synchronization of theta rhythm during its running, called theta rhythm. This is observed local feed potential, like a leaked potential, not directly recorded from one single neuron. A kind of summation. It's a weak signal, but it's a kind of the wave. Cell 1 fire if the rat is located here. That's why cell 1 fired if rat is located here. If rat is located in the green area, oh, start to fire cell 2, fire, 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 during that is located in S area. Third places, if rat is located here, so 3 fire, 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 fire. Such a firing timing, initiation are different because the rat located in different way depending on position. Okay, this is a kind of a simple explanation. Place cell. Place cell means that the cell is fired depending on the place. This is a kind of the activity initially O'Keefe discovered. Finally, O'Keefe also got Nobel Prizes according to Presto. Skaggs is the founder that discovered the specific firing pattern with the supervisor Bruce McNaughton. I really loved the Bruce McNaughton. I climbed the mountain top of the Mount Fuji with him, by the way, with his wife. Theta phase precision, specific type of the firing pattern. Then not only the specification of location, but also timing. Okay, you can notice as the timing of the spike with respect to the theta rhythm phase. Fire here, fire here. Okay, if firing timing gradually shift more earlier, which means the firing, the bit shifting the location of the phase. Such a systematic firing occurs consistently with different cell. If cell 2 fire in this here, cell fire, 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 gradually the phase is shifting. Cell 3 fire suddenly here. So then you can focus some sequential structure. If you can focus some one window of the theta rhythm, cell 1 fire earlier, cell 2 fire secondly, cell 3 fire third places. So which means that in one single window of the theta rhythm, cell 1, 2, 3 sequentially occur. This is quite interesting because a plasticity means change the synapses, strength of the neuron to neuron. It's the basement of the learning capability. So in the hippocampus, specific type of the synaptic plasticity exists. It's called Hebbian plasticity or asymmetric Hebbian. Spiking dependent plasticity is very interesting because the firing red one fire next just after 20 or 50 milliseconds post firing fire, red to blue, fire 1, fire 2 is occurred. A snap plasticity generated. Then red to blue, the connection is strengthened. But the negative order, I mean the blue to red, is synaptic connection getting decreased. Finally, it represents red to blue, okay? You may understand, okay, the hippocampus have the function of the episode, similar like a song, okay? If you hear the beginning of the song, you can rephrase, okay, this. You can sing a song because the initial cue. The episodic memory is also similar structure. If you get some initial cue, your memory retrieved sequentially. This is coming for neural network. The cell 1, 2, 3, 4, 5. Okay, like a cell 1, 2, 3. So this is a very interesting structure this systematic sequential activity generated synchronization with different phase is connecting such a prosody, the time window of the prosody. Therefore, this micro phenomenon is connecting such a systematic firing pattern, one top level, and generates asymmetric connection with the neuron. Then it records episode or sing a song or playing instrument. This is the amazing thing. Therefore, okay, the music happening inside the hippocampus. 1, 2, 1, 2, 1, 2, 3, 1, 2, 3, 1, 2, 3, 1, 2, 3. This kind of sing a song is happening inside the hippocampus. This is a kind of amazing. This is a metronome. The normal one. The interesting phenomena, artificially generated. You can test by yourself. I do the same thing. Even a pet bottle experiment. Can you see this? What's happening? It starts randomly, different phase. But you can see basement is shaking like a local field potential. Now it's getting synchronized. Yes, yes. This is also mathematical solution if you solve differential equations that are the pendulum. This kind of activity happening in neurons, and the neurons model are the network. Now it's totally synchronized. Therefore, yes, you can describe this kind of network by mathematics. So then Yamaguchi and McNaughton published this paper, and the initial the formulation of the mathematics to represent such a phase precision mechanism. This is differential equation, some specific parameter of the basement frequency and some coordination parameter. If okay, you can imagine athlete, runner in a track. Runner is running in the circuit. Run, 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 run. It's running. So then newcomers entering the new track, this track, then this. Okay. If red is input is happening in the red cell, start firing, frequency gradually increase, and the firing phase gradually shift. Then blue one is coming into the next. So then frequency gradually up, and firing phase gradually shift like this. Then it clearly generated this mathematical circuit, the differential equation clearly reconstruct the phase position. Synchronization, but different phase, systematic shifting. It was so amazing. The same activity is coming from the nervous system of fish, but also snake, because such neurons embedded the backbone inside. Then such a firing pattern happening. This is the control mechanism of the body. But interestingly, according to the evolution, this same function is used not only the motion control, but the cognitive function. Now our brain has such a systematic firing pattern originated from the motion pattern generation. In the internal cortex and hippocampus, then the systematic firing appear our brain like a sing a song and playing instrument. Then generally we can remember what you're coming from, who you met, what kind of food you ate before. This is the episodic memory. Then I will show you again, they are such amazing snake behavior. So this is a big mystery. I show you again, this is a motion coming from such a pendulum, okay? Such a synchronization of the neurons fire together but a shifting way. Then snake behaves in consistency like this. This is a very interesting things, okay? The nervous system again, even if the motion of the cat jumping, also coming from this kind of synchronization phenomena. That's quite amazing things. So back to the episodic memory. It was my topic in my PhD season. Okay, this big controversial issue exists in that field. Both are professors. The famous person debated very seriously. Howard Eichenbaum. Yes, they cannot deny firing pattern because its existence of the specific firing pattern is hippocampus or rat. But Howard Eichenbaum is focusing on episodic memory in the human. That's why at the lack of the hippocampus, we lose any of the episodic memory. That's why Eichenbaum think, okay, functional hippocampus not only for place representation. That's why place cell maybe yes, but spatial map is no. He denied the hippocampus at the brain region only for the managing spatial representation. This is a part of the action, they say. That's why there's a place cell activity exists. But John O'Keefe say, this is very specifically designed as a spatial geometrical function. That's why hippocampus have the basement of the consistent cognitive map solely. Different cell represent different location. That's why hippocampus focusing on the spatial information. Theoretically, what kind of problem happening more precisely? If Eichenbaum is right, episodic memory should represent asymmetric connection because of the history, okay? 1, 2, 3, 1, 2, 3, 1, 2, 3, sequential memory. But if it's a spatial memory, network represent a kind of distance. This node is close. This node is far, for example. Therefore, very simply, okay, non-directional. No, the directional. The IJ is not the equivalent. But this IJ, the JI is equivalent in the case of the non-directional. The connection is connected if it such a linkage that represent strengthen the neurons represent distances. Okay, symmetric connection is right. So the controversial the point of the view that initiated from the Eichenbaum and the O'Keefe in the form of the mathematics, we need to think about the contradiction of the asymmetric connection versus symmetric connection. So we need to check such a the theoretical background review or the other paper. This is the point of the view now. So then I try to reproduce similar kind of activity. This is the actual data. The neuronal pattern, the SKAX obtained. Some specific pattern at the population, this activity represent the location. But some kind of special activity happening like this. This is a computer model. It's my design. If feeder precision, firing sequentially, occasionally firing synchronously, but a different shift, like 1, 2, 3, 1, 2, 3, 1, 2, 3, such a sequential activity happening. According to the position red, this activity have some specific tendency like propagation. Okay, you can see actual phenomena in the brain, but my computational model also shows such kind of things. Okay, it represents, okay, firing pattern not only in the location, but also wave pattern. Wave pattern distributed, propagated forward. This is the publication in my paper that collaborated with my supervisor in that moment. That's why I go to the PhD. This is quite new for this. Because of the focusing on the controversial issue, Eichenbaum and John O'Keefe, I explained the mechanism. If rat is learning single way or multiple way and spatial way, the neural network have a specific the connection pattern like this. This is coming from the wave pattern. That's why some asymmetric the structure happening in around the cell. Wave propagate in different direction. Then finally, such a mountain-like the connection is formulated in the sense of the spatial representation. This is the actual the hint to understand why these two different type network is existing. More seriously, I analyze such a the connection pattern is even after the spatial exploration, that the centralized the mass of the center of the weight is not simply located at the same position in the cell. They are shifting a little bit. Therefore, such a some specific asymmetric connection exists. In that sense, Eichenbaum's right, but also in the global structure, O'Keefe is also right. Because such the microscopic and the macroscopic two-level representation occur, one specific phenomena inside the brain, especially hippocampus. This is a computer simulation. I generally designed this computer simulation in my PhD period. So newly established from differential equation. So this arrow represents such a directionality for the cell the connection. Finally, as a global structure, if arrow is aligned same way, the asymmetric function exists. That is it appeared. But arrow is distributed different way. So then they cancel together. That's why spatial activity happening. This is the summary of the paper. That's why single way and directional structure globally remains. But according to the exploration, such a geometrical structure that change differently. Then finally, they represent spatial portion. This is also experience-dependent population activity is generated computationally. Entering cortex that receive information rat can see from so many cues, and then propagation activity start, and then retrieved in the sense of the CS3 local connection. Then they can predict what is the next, what is the next. If it's after the short run, they can predict forward. But after spatial exploration, they can remember so many items in surrounding places like these. This is the solution of the big mystery in the past. Why such a controversial issue is happening? It's not coming with a simple manner, but complex manner, but the principle is very simple. Therefore, even the one short moment, the single cycle, the theta rhythm, current perception, pattern completion, and anticipation happening. Therefore, I hypothesize that moment, yes, our brain, okay? During your walking, you see some cue in front. Your memory can retrieve and anticipation occur. Every single cycle, the theta. Okay, let's say 0.1 second. Every 100 seconds, you sing a song. Your brain sing a song. Repeatedly, your imagination again, comes back again and again and again. This is a function of hippocampus. That's why repeatedly happening. The episodic memory, then the connection is established. This is the first robotic platform I built it alone in the Riken Brain Science Institute. This is the kind of initiation, neurorobotics in that moment. I reproduced a small robot, then such a recognition happening, then a local brain simulator works well on the computer, then reproduce same way. So then I would like to summarize the fourth topic. So to know the best timing to do, like driving the car, we have a type of the memory like this. I'm focusing on the explicit declarative memory, episodic and semantics. Motor function is very important, of course, but we are using both together. Explicit, explain your conscious level. Oh, car is coming. It's not good guy. That's why I cannot allow enter. But you also control your body at a very implicit way. That's why the brain, explicit memory and implicit memory work cooperating together. I would like to summarize again types of memory. Okay, short term, long term. Now I'm focusing on the explicit memory, especially in episodic event. So this is the moment, 2014, Nobel Prize. I was so surprised because my work is the kind of, okay, John Lec4 found that this discovery very old time, but finally, the couple, the mother, also found such a great cell structure of the rat brain. Finally, that the work is evaluated in a sense of the Nobel Prize level. And then Nobel Prize in the Physiology and the Medicine 2014, then John Lec4 got Nobel Prize. I'm so proud of that. Of course, this is the initiation of my PhD work in that point. Recently, not also recently, the IBM or other company also they're interested in the neuromorphic chip. Now, in my department, Professor Tanaka or Professor Morier and Professor Tamko, also they're focusing on the such a neuromorphic chip too. This is very interesting. The performance is comparison to the conventional type of cell. This is kind of the artificial neurons embedded in the circuit and connecting neuron in the bus, the synapses in the neural circuit. Then they try to reproduce similar kind of the function of the brain. Then, so very high specs resolution reconversion, the image processing is done. If it's low resolution, people cannot be recognized. But such as the motion, what where pathway in the brain, the person in the cyclist can be recognized by chip very fastly. Okay, this is the today's second topic. It's in the fourth lecture. Is there any question from audience? No? In chat? Chat? No? Okay. Did you enjoy? Okay, show me the reaction. Did you enjoy my lecture today?
Yes, sir.
Okay, thank you very much. Okay. Okay, in this week, I completed the 4 lecture and then also the prepared the KWM. Is there any problem in the KWM assignment now for this participant? You completed? Okay, so I really hope to see your comment. Okay, I can reply to you. Okay. Okay, if there are no questions, okay, I would like to close. Thank you very much. Your long patience of hearing my voice. I try to improve my system, but okay, limitation on the internet. You may, I'm not sure that you're hearing my voice in the good condition or not. Okay, but I really hope, okay, you enjoyed my lecture. See you again the next week. Okay, or we can discuss about the issue in the educator system. Okay, happy weekend.
Thank you, sir.
Thank you, sir.
Thank you very much.